I was talking that I will be talking in this session about biological networks, and this is mainly a domain or one of the disciplines under systems biology, right? So I will be talking about different networks that are available in biology, including protein-protein networks, and we have this gene-gene networks, metabolite networks, signaling networks. So these are different types of networks that are available. And I will also show you how you can analyze this using available tools. One of them is very popular called Cytoscape. So I will also show you some demo on Cytoscape. So before that, let's start with knowing about what is system biology, right? So we know that our human body basically is composed of, or we can think of it as a whole system, or we can also have systems. We can think of systems at different levels. So, for example, we can have network of organs, right? We can have organs that are connected to each other. We all know that they are all somehow interconnected. So one can think organ as also as a system, or one can, as I was talking about nowadays, single cell in previous session, single cell or cellular networks can also be from one another layer of system, and then we can have molecular networks within this system, or we can even think genes as our systems. Even proteins can also be considered a system. So basically when we are talking about networks, so we have most of the things in biological networks, they have been adapted from these social networks, where we see that a lot of social platforms like Twitter or Facebook that we see a lot of how we see the connections and how people are interconnected in a social network, right? So we have, let's say, we call this the central or hub node or hub person, a very important person which is connected to so many individuals. So similarly, we can have protein networks, or we can have gene networks, and we want to study which genes are interested or which genes are important in given network. So particularly if you say that a gene like this is connected to many other genes, so probably this gene is important or performing some important function, biological function, to say. So what is system biology basically? Systems biology basically, there is no as such, there is no definition that describes this field, but it is also used in many contexts. It is used in several different many contexts. So a lot of definition, but nobody is really clear what do we mean by this terminology, systems biology. So this was mentioned by one of these great scientists, John Yates. And even if you look at the textbooks, even there are no clear defined textbooks also for this field. You cannot have a single book which describe, or we can follow at present to which describe all the aspects of this field. And you will see that in internet, if you look for, you will find many definitions, but basically you can say it's study about systems. So there are many things that are at the heart of this field. One of them is the modeling process, and most of our computer science people are very well aware of this. But let's understand it again with the help of this cartoon. So we have basically, in general, we can categorize this whole world in terms of let's say real world, and we can have another world that is called math world. So in real world, we have real world problems, like we want to study the function of some gene, or we want to identify important gene to say in terms of biological questions, or we want to understand which genes are important, or which sequences are coding for which genes, so things like that. So we can have this kind of problems in real world, or we can even have some layman problems like from where, what is the shortest path from one place to another problem, to one place to another place, or one city to another city, right? So these kind of problems exist in our real world. And we always, when we start with the modeling process, we most of the time we start with the real world problem, right? And then we need to then we enter this math world, where we need to define this real world problem in terms of a math problem. So probably we have to come up with some equations and some variables, parameters that will describe this real world problem in terms of mathematical equations. And then obviously we will be like making some assumptions, and we will probably start with simpler assumptions first, and probably we will come up with some set of equations that we will be able to describe, or to describe this real world problem, or we can do some predictions, or we can describe this problem into. And the next, what we do is we solve this set of equation with the help of available analytical or numerical approaches, and then we come up with a solution to model this behavior, or we come up with a model finally, and then we do some validation with the help of external data available from real world, and then we probably draw some implications and inform about this problem. Some solution is provided, and this cycle can be also be repeated, and even within this we can have some repetitions, right? So we can have some iterations here, and this is how you can say very abstractive view of a modeling process. And to be more specific about at each level, so we can have so usually what we have is different stages of the modeling process. We can define it in terms of like this. There can be other several possible ways, that is definitely possible, but usually we have our goals, inputs, and our initial exploration of the problem, and then we have model selection, and then model design, and then model analysis and diagnosis, and then model use and applications, right? These are all different major steps, and within these major steps we can have several sub steps, or we can say very other small steps. We can have, for example, under the goals inputs, we can probably what we will do is we will define the scope, what are the objectives, what are different of this process by we want to do the modeling, what is our objectives, basically defining our real life problem, right? So, and then we need to collect the data and then information, and probably we will also gather some prior knowledge on this about this problem. And then we come up to the next step, where we have to think about model selection. So we need to think about what kind of type of model will be more suitable, whether it is a dynamic model, static model, correlation model, or mechanistic model. So there can be several list of possibilities. Once this is done, probably we have to then next thing that we have to do is we have to then decide what are the variables, interactions, and equations and parameters and other things that are available, and that we need to define in order to make the model or design our model. And then next what we have to do is we have to perform some model analysis and diagnosis. So we will probably check for many things. We will look for consistency, robustness of the model. Then we will also look for validation of dynamics, exploration of possible behaviors, and so on. And you can see that these are itself are iterative process. So you may have to go back and probably throw or add new variables into your model, and again come and check all these steps and perform this model analysis and diagnosis steps. And there are many methods to do that. So we will not be looking into those methods in this session, but there are several approaches for checking all these parameters or all these things, not parameters. Then finally, let's say we have come up with a good model that looks good, at least on the training data. Then we come up and to application, right? Where we can apply this, or where we do some validation. Sorry, in testing, you can say, where you can test, for example, some hypothesis, or you can use it for simulation, or you can do some prediction or explanation, and manipulation and optimization are the next steps after by using this application based knowledge. And then maybe you have to go back and also repeat these steps again. Now coming to the networks. So we have, as I was saying, that networks are basically a collection of nodes and edges and connection between them. So nodes can be anything. So you can have genes or proteins, or a major topic that we have in system biology is, as I was saying, is networks. And then these nodes in the network can be genes, so that makes up gene network. And if we have nodes as proteins, we can have protein interaction networks. Then we can also have metabolites as our nodes, which will give us the metabolic networks. And then we can have, similarly, we can have these signaling pathways that can also be represented in the form of signaling networks. So biological networks are of different types, and different theoretical methods are already available that can be used for different types of networks, right? So these are all existing methods that have been adopted in biological networks. And the individual networks, for example, gene network or protein network, they must be unified to get an understanding of the system as a whole. So we can, again, it comes to the concept of integrating different layers of data. So you can think gene networks as your genomics information, protein interaction as proteomics level information, and probably you have to again unify. You need to have a unified understanding of this in order to understand this system. And there are also simplified mathematical models that can be used to study these biological networks. And there are also complex methods that are available, and also there are computational models that are available to study these biological networks. So many of these models are already available, and they can also be used. And I will probably also show you some other in some of the slides how you can what are different models that are available. So this is how this, let's say, protein network of Drosophila looks like. So this was published in Science in 2003, and it has become like one of the most attractive figure in the field of protein networks. And you can see, so most of these biological networks are really very complex. And you can see this complexity of this network, this where you see there are so many thousands of nodes within it. And you can see if you want to like have an overview or zoomed, if you zoom in these regions, you will get that there are many possibly interesting things that are happening within these regions. So for example, you can, if you zoom in, you can find which kind of proteins are interacting with each other. So it is basically protein interaction network, which means that if there is a functional interaction or physical interaction between any two proteins, that will be reflected by the strength of this edge that is shown between these proteins. And the legends basically describe the subcellular localization of this protein. So which regions of the cell these proteins belong to. So some may fall into extracellular, or maybe you can say plasma membrane proteins or mitochondrial proteins. So this is very, you can say, an interesting paper that have come up in early 2000. And you can say, yeah, as I was saying, this the thickness of this edge will represent the strength of interaction. Now let us discuss about some few of the properties of these networks. So most of you, those who have learned graph theory, they are familiar with these terminologies. So one of the important, so there are many properties of the network, but I will just take you through only those that are mostly relevant for biological networks, in the context of biological networks. And so one of them is degree centrality. So basically what does it shows is it shows how important a node is, and degree, higher the degree, higher is the centrality, or higher is the importance of this node. And if you have higher, if a node is having very high degree centrality, that node can be considered as hubs, which means that they are connected to many neighbors. So degree is basically the number of immediate neighbors, or first degree neighbors of a given node. And there are different ways to describe it for directed and undirected graphs, or directed and undirected networks. Basically, undirected networks, we do not consider directionality between any two nodes, whereas in directed graph, we need to also consider the directions. So in biology, there are both kind of networks exist. So for example, if you look at this gene regulatory network that I will be talking about, where you will have this direction between the genes. So if there is an edge from gene 1 to gene 2, then it means it need to have a direction which will tell us whether gene 1 is activating or deactivating gene 2, right? So this kind of information can be seen in gene regulatory networks. But in case of protein interaction networks, which basically it doesn't make sense to have a direction, because if I say that protein A is interacting with protein B, it also in means that the protein B is interacting with protein A. So here the directionality is meaningless. So we have both type of graphs that are networks that are available, directed as well as undirected networks. And so degree can be calculated differently for these different categories of networks, directed and undirected ones. So most of the, I think in this session, I will be talking only about undirected graphs or networks. So we can have another measure that is called closeness centrality, which is also one of the important parameter. Okay, so this basically indicates that we can have it will indicate this important nodes that can communicate quickly with one other nodes of the network. So how close it is to the other networks, and it is used to identify the top central metabolites in genome-based large-scale metabolic networks. Right. So if a node has high closeness centrality, means it is probably communicating with the many other nodes that in the network. And also there has been studies to compare unicellular and multicellular eukarya based on this closeness centrality property. So there are some studies that have been done. So though these properties are very simple, but they have been like used like anything in biology, and they have given many informative insights in solving many biological problems. And so another property that has been extensively used, has been extensively applied or to study biological questions, is the betweenness centrality, which actually shows you that that the nodes which are intermediate between neighbor neighboring nodes will rank higher. So basically we can have betweenness centrality for a node, or we can have betweenness centrality for edge also. I will show you in a coming slide how you can use this measure, a little bit more information, because this has been it has been found to have many applications. So people have have found proteins which have high betweenness centrality, and they have been termed as the bottleneck for their role as the key connector proteins with essential functions and dynamic properties. So this is just one observation that I'm showing you, but there are many other findings that have also been noticed, which rely on this betweenness centrality measure. So let me tell you a bit more about this. So as I was talking, that betweenness centrality can be calculated for a node, or it can be also calculated for an edge. So how it is defined? So it is defined as the number of shortest path. So this is just I'm talking about the edge, but it can be also it can be applicable, or it can be just modified also for the betweenness centrality for a node as well. So for in case of edge, it is defined as the number of shortest path that go through an edge in a graph or network, and this was proposed by Girvan and Newman in 2002. And each edge in the network, we know that it can be associated with an edge betweenness centrality value. So we can calculate for each edge in a network, and we can also calculate it for the whole network, this property. So an edge, say in this figure, you can see here in this network, let's say we have this edge E prime. So we are expecting that this will be having a high edge betweenness centrality score, because it represents a bridge-like connector between two parts of a network. So if you think of the whole network can be think of made of two communities. So this subgraph and this subgraph, they are connected through this edge E prime. So you can imagine that this even this E prime edge is of high importance, because it is connecting, or it is having it is connected, or it is you can say it is making a bridge between two subgraphs in this whole network. So if we remove this as E prime, so we will it will affect the communication between many pairs of node. So for example, any of these node will not be able to communicate with these nodes if we just remove this edge E prime. So this there are computational so there are mathematical formulas that have been provided to calculate the betweenness centrality, and primarily it is also now it is being provided by Girvan and Newman as a form of algorithm. So algorithm starts in this way. So you break, so you calculate the betweenness centrality score for each edge, and then you remove those edges, the edge which has the highest betweenness centrality score, and then you recalculate this you recalculate this score again for each of the remaining edges, and so on. So this is iteratively iterated until you are left with none of these edges. So only the nodes are left, and then you then reconstruct this network to in the form of probability in the form of this hierarchy in dendrograms, to see how you can if you are able to just form this network again. So this algorithm is very well known, and it has many applications even in biological field. So for mathematically, if you want to define this for a node, so this is how it is defined. Betweenness centrality for a node V, let's say for a node V will be given by the summation of these fractions sigma st v, which is basically the number of shortest paths from node S to node T through that passes through this node V. So this is the number of shortest paths. So shortest path basically means that what is the shortest path from node S to T. So how many edges you have to cross in order to reach from S to T. There can be multiple paths, right, from between S and T, but we only include only those paths which are shortest. All right, so we can also have total number of, so in the denominator, we have total number of shortest paths from node S to T. So this is basically the fraction of these paths that pass through V and those do not pass through V. And then we can just take summation, and that will give you us the betweenness centrality of for this node. So just to show you an example, so just this is just one network, very small network you can think of it as. So you can think that you can just by looking at this network, you can think that you will say that yeah, V1 seems to be very important node because it is connected to many nodes and many paths that connects between any pair of nodes that will pass through V1. So for example, if you want to go from V7 to V5, you have to go through V1, and if you have to go from V7 to any of these nodes, then again you have to go through V1. So no doubt, just by looking at the V1, just looking at this network, we can say that V1 is important, probably the hub node. So this is following the previous slide. I am giving you these measurements for betweenness centrality for each of this node. So for G1 we can see that betweenness centrality is 12. For G2 it is 8, and for G3 it is 5, and for G4 and G5, G6 and G7 it is 0, because these are peripheral nodes V6, V7, V4 and V5. So we do not expect any paths going through these nodes. If we want to go from V1 to V2, probably we don't have to go to V7 and V6 and V5. So that's why their score is 0. Betweenness centrality score is 0. And also, as I was saying, so you can see that this betweenness centrality score for node 1 is highest. Okay, so there are some other additional features of networks. One is the called as the connected components. So in case of undirected graph, basically it represents a subgraph in which each pair of nodes is connected with each other via a path. So there exists a path between each and every pair of node. So in case of like the example I showed you back, so this can be one subgraph, because you see there is a path between each pair of nodes between here to here, and then from here to here, and here to here, here to here, and then also any of these pairs can be reached from any of the node. And similarly, so this cannot be a subgraph because there is no path between this node and this node. So we only have one subgraph. Or sorry, not subgraph, but a connected component. So in connected components, all the nodes are reachable from each other. So this is what I was saying. And so there is another definition for directed graph, which is very similar to connected components for undirected graphs. So we can have a strongly connected component in case of directed graph, which basically the definition is, again, it is same, but slightly different than undirected graph. So there is always a path between every pair of nodes in a component in both the directions. So here we need to have the path between any pair of node, every pair of nodes in this component in both the directions. So the direction is important in case of directed graphs. And then we can also have cyclic graphs, which is basically a graph which contains at least one graph cycle, and there is a path from a node to itself. So this is the definition of this cyclic graph. Then another important definition is motif. So I will talk about more details in coming slides about motif. So basically it is a repeating occurrence of simple patterns. So what is motif? So motif is basically a simple patterns, right? So we have patterns which are more frequently, or you can say statistically significantly overrepresented in subgraphs. So motifs will be, you can say, they can be also subgraph. So for example, I will show you that there are different types of motifs that exist in biology. So it can be coherent feed forward loop, or where we have feed direct and indirect feed. And then another concept that is very important in context of biological networks is a random walk. So a random walk is basically means that in a given on a graph, it means that you are starting at any vertex, at some vertex, and then you are traversing the graph according to some probabilities. Right? So so each time you start with the random node, and then the probabilities are assigned to jump from one node to another node, and these probabilities makes it a random walk, because you can take this way or that way depending on the probability of going between the nodes. Again, I was talking about this degree, because degree is one of the important parameter measurement that is used in biological networks. And again, this is just I want to show you some models, so that's why I am just giving you a bit more formal definition. So degree is of a node in a network is the number of connections it has to other nodes, and it is usually denoted with the small k, letter k. And we can have degree distribution. So degree distribution is usually described as p of p to the subscript k, where we say that degree distribution is the probability distribution. It can have different types of distribution that are possible, are binomial or power law of these degrees over the whole network. So how these degrees are distributed, right? For a given network. So below in these figures, you see some examples from real biological networks. So this network, for example, has binomial degree distribution. So as you can see, right? So the distribution looks like binomial distribution with an average number of average degree for this network is around 10. And so I have not just put in, I think it has around 100 100 nodes. Then we have this another type of distribution that is possible, that is power law degree distribution, where you can have the distribution of this degrees looks like a power law distribution. You see there is one peak over here, and then it just decaying like this. So what is important or what is peculiar about this power distribution is that you only have a few nodes with high degree. Most of these nodes, they are, you can say, they have very low degree. Right? So only few hub nodes are available. And interestingly, most of these biological networks, they follow this power law degree distribution. And I will also talk about this in coming slides. So where we will study about different models. So what are different network models that have been proposed and they have been used in the context of biological networks. So one of them is Erdős-Rényi model that basically describes the random networks. So Erdős-Rényi or ER in short model, they describe random networks where the degree distribution is given by binomial distribution. So if you think of a random network, basically that means that there is the probability. So there this the probability of connection or between of an edge between two nodes is random. So what you can imagine it like this. So suppose you are given n number of nodes and let's say e number of edges are given. So what you will do is you will just throw these edges on these n nodes, and it will randomly be assigned, or it will randomly be connecting different nodes. So each time you throw these edges, so you will find that each time you are throwing these edges, you will get a different structure of the network. So this structure is random because each time you are throwing the edges, you will the chances that it will connect, there will be connection between any two nodes is random, right? So it is governed by some probability. So you can assign some probability, and you can then throw that these edges, and then you will based on what kind of probability you have assigned, you will see different sorts of an ensemble of networks basically to say. And then another set of model that has been very extensively used in terms of networks in general is Watts and Strogat's model, which describe networks that follow this small world topology. Means that most of these small world networks that we see, they are you can say they can be modeled. So for example, social network, or you can say our real life networks that exist, they can also be modeled in this with the help of this Watts and Strogat's model. But in biology, I think the most of this network, they are modeled with this in using this model, Barabasi and Albert model. And there is also a nice book that that describes this these real life real world networks, where we where they show that there is 6 degrees of connection between any two individuals in this world. Means that there is probability that you know every sixth person in this world. And so this is how this model also describes. So it describes these networks in those that follow the power law distribution, because most of these networks, they have this low degrees, whereas only a few of these networks nodes are highly connected, or they have the very high degree. Also, if you think it in terms of like real life, most of these real life networks small or also kind of follow this kind of distribution. Because if you think of, let's say, social connect, let's say Twitter networks, they are also like only a few people have a lot of connections. You to say, let's say, if there are any celebrity or any political person, most of they are probably connected to many nodes rather than in general like normal people. So and also in biology also you will sorry, and what I want to say is that in biological networks also this kind of phenomenon is very frequently observed. So so one of the challenge or one of the problem that is usually discussed is identifying the communities in the networks. So you will see that how many let's say which genes or which proteins or group of proteins that are involved in some biological pathway, or they are involved in some disease mechanism, or somehow they are interacting, or they are always seen together in biological networks. So Communities are basically these groups of genes or these groups of biological molecules that occur more frequently than expected, and there are different ways that has been proposed to identify communities. These are the three more methods that I am listed here. I have listed here, but there are so many algorithms. There are so many algorithms from graph theory, such as click identification, maximal spanning graph, or you can say maximal maximal modularity and things like those. They have been described very extensively now in this by this time, and clustering and also Griman and Newman algorithm is more popular in biological networks, and I have already talked about these algorithms in terms of betweenness centrality. And there are also modularity algorithms that are available. So what is modularity? So basically a modular system is a system that can be, you can say, decomposed, that can be decomposed and at least to a first approximation into structurally independent parts. So you can think of it has like some communities that exist, or they act, or they function independently. And structural modularity of a network can be quantified with the help of there are many algorithms. So for example, you can quantify also with the help of Newman Girman algorithm, and you can also use other algorithms. Of course, there are many other, and you can find these algorithms attempts to partition this network into parts, or you can say communities, such that these parts have more connection inside them rather than outside. So there are more inter community or intra connections than interconnections. And of course, this degree to which a network can be decomposed in such a way can be used to provide a modularity score. So there are different ways. There are mathematical formula that can be used to provide modularity score to the networks. And most importantly, so if you think this, is it possible that random networks will be modular or non-modular? So yeah, of course, because random networks, we will not see many of modularity. They will be mostly non-modular. And in context of biological networks, you will see, or even biological system, you will see at each of the scale, you will see there is high modularity. So let's take some examples from biology, so where you see this modularity. So for example, you can think our body, as I was showing in the first slide, you can think our body is made of different organs, and each organs is a module because it has some special structure and it performs specific functions. For example, heart is used for breathing and you're pumping the oxygen. So it has specific functions, right? So liver has its own functions, kidney has its own functions. So these organs can be considered as different modules, right? Even if you think, just if you go to let's say nanoscales, or even if you just think, proteins also can be considered as systems itself. So for example, proteins, you can think them as made of modular domains. For example, typical transcription factors such as this is just one example, lac repressor lac, which has different domains. So proteins, as I was saying, it is basically a sequence of amino acids, and within this you will find certain domains or set of, you can say, a subset of nucleotides, subset of amino acids that will be performing some specific functions. So for example, one subset of this amino acid called domains can bind to another protein or another molecule and another one domain can also have another function. So basically the idea is here that protein has different domains. Different domains can perform different functions independently. And of course we cannot draw a line between, we cannot say that entirely independent, because biological systems are also communicate, and there is also a lot of communications happening. So yes, so to some extent they are modular, we can say. So I was talking about network motifs, which are very interesting to study in context of biology. So as I already explained, that motifs are basically what are patterns, right? So these are patterns of connections within a network, and patterns that occurs in the real network significantly more than often, they can be treated as the network motifs. And you can take some examples of motifs. So examples that consist of only a single edge, right? So you can think of a connection between two nodes, and also these are directional. So you can think of that this node A, node B, they are connected, or they are, this edge is found more frequently than in a random network. So you can look for those kind of connections into your networks. Or also importantly, you will also be you will be also interested in studying these self loops, so self arrows, and these are called autoregulation. And such kind of phenomena is very well known in biology, where a gene, for example, control its own expression. So a gene is being expressed, and after a certain level of expression, it stops. It stops expressing by, like you can say, it controls its own expression. It just shut off at after a particular threshold is reached. So these kind of motifs are also interest to in biology, and a lot of work has been done also in this context, and many algorithms have also been found to study these autoregulations. And another example of motifs is involving two different edges. So you can have a reciprocal motif, where you see that the gene A is regulating gene B, and then gene B is also regulating gene A. Right? In contrast, here you just have gene A regulating gene B, but here you have reciprocal regulation also. Right. So these kind of motifs are also of interest, and then you can have convergent and divergent motifs, and you can have chain motifs. So these are all possible motifs with two edges. And then there are also other motifs involving different other edges, like you can have motifs for three edges. So the complexity also increases as the number of edges increases. That is very obvious, right? So the natural question now, probably we have to that can cross our mind is, how do we identify or detect these motifs? So one possibility is that we compare these network motifs by comparing to randomized networks. So one thing that I want to tell you is that this, when you are saying random network, so basically this is not a single network. So this is a ensemble of networks. So when we talk about random networks, we can have, it is an ensemble of networks. We have thousands of networks that you categorize as a random network. Right, so let's have a look on how we can use this to identify or to detect network motifs. So for a meaningful comparison, the randomized networks should share the basic features of the real network. Right, so this is one of the important condition. And this real transcription network, for example, will have, let's say, n nodes and a number of arrows, right? So to compare it, remember Yadav's Rene model is used for random networks. One need to build a random network with the same number of nodes and arrows, right? So we need to make a equivalent random network to our real network, where the condition is that we need to have the same number of nodes and same number of arrows. So for example, if our real network has n number of nodes and a number of rows, you need to have the same number of rows and nodes in your random network. So in the yeah, so as I was saying, so this ER model, so arrows are assigned at random between pairs of nodes. So with the random means there is some probability. So let's take an example. So you can see that on the left side here, suppose this is our real network. So we have created this network from biological data, right? So we have in this network, we have 10 nodes and we have 14 arrows. And number of n self, that is basically the self arrows, or you can say this kind of auto regulations. So this is our real network. It has 4 self arrows. So now how do we study, or how do we create this randomized network? So as I said, so we need to have not only single network, random network, it will be ensemble of network, will be implying that we will have thousands of such networks. But the condition is that we need to have the same number of nodes and same number of arrows. So each time we throw these 14 arrows on these 10 nodes, there will be a different structure that will be observed, right? And then what we will do is we will create the distribution. We will check where, for example, we can check how the degree distribution looks like for these random networks, or we can calculate, let's say, different measures from these ensemble of random networks, and then we can perform some statistical test to compare this our real network to the random network distribution. So this is how we can use this random networks approach to identify motifs. If the motif is significant, then we will observe this more. You can say in case of random network, it will not be that much significant. So it need to be significantly observed. So basically what I'm saying is these motifs are statistically overrepresented in our real network. And so I was talking about this autoregulation. So this is, let's take an example of how we can study this autoregulation with how we can detect this autoregulation with the help of random networks. So suppose you have this simplest network motif, as I said. So this autoregulation is simplest network motif. And example and biology is, of such kind of regulation, you can see it terms of transcription factors, which are primarily the proteins or genes that regulate the transcription or expression, you can think of their own genes, right? So is autoregulation significantly more frequent in the real network than at random? So this is our question, right? So to calculate the average number of self arrows in an ER random network, so you can think of it how we can calculate this average number of self arrows in a corresponding ER random network. So to form a self arrow, what we have to do is but we have to draw an arrow. Arrow needs to choose its own node of origin as it is destination. So it starts from a node and it comes back to the same node. So origin and destination is same. It's the same node for a self arrow. Now let's say let's say if I start from a node, and if let's say if I, so how many possibilities are there to draw an arrow? Right? So we will have n different possible targets. If we start from any node, from a given node, and if there are n nodes in the network, so we will have a probability of self arrow as 1 by n, because each of the node has got equal chances of connecting from this node. So if we start from this node as origin, so we will have 1 by n probability of coming to the same node, or to the destination is also same node. So probability of a self loop, you can say, or self arrow, is 1 by n, where n is the total number of nodes in the network. Now since we have, let's say, a arrows are placed at random to form the ER random network, so the average number of self arrows is a times p self, right? Very straightforward. We just calculate it by multiplying the probabilities. So a by n will be the expected number of self arrows in a random network, right? And then we can also calculate the standard deviation, which is given by taking, which can be calculated by taking the square root of this n self. And now what we can do is we can just compare. So let's say if we take an example of this network, so we can calculate, we can do some calculations. We can calculate how many expected number of arrows are there in this random. So we have n is our 10 and a is our 14. So by following up this, we will get 14 by 10, that is so n self, expected number of self is 1.4. so that is basically 1. So expected number of n self arrows is 1 in case of a random network with that number of arrows and that number of nodes. And the standard deviation can also be calculated. So again it will be square root of 1.4, which will be close to 1 again. So for this network, if you think of whether this is significant or not, you can check, okay, for random network, yes, of course we see n self is 1, this is what we expect. But for let's say for this real network, we have n self of 4, which does not change, which does not seems to be that statistically doesn't seems to be that high. But let's say if this number is if in your real network, let's say you see that n self are 25, so then you can think that, okay, yes, this n number of this n self is statistically significant compared to the random network. So this is just one artificial example that I am showing you, but this can be applied to real networks. Then I was also talking about, let's say if you have more number of nodes, and you can have other possibilities of defining the motifs. And one of the interesting one is this feed forward loop and feedback loop, right? So where you see this direct and indirect feed, and here you can see a